How to make an endospore stain. Step 1. Turn on the bacticinator. It will take 10 minutes for it to reach optimal temperature. For this experiment, you will need endospore-forming bacteria. This culture was made on a spore-producing enriched medium. A clean slide. The primary stain, malachite green. A secondary stain, saffron dye. A clean paper towel square cut to roughly the width of the slide. Bibulous paper for blotting. A hot plate. 140 milliliter beaker. A sterile inoculation loop. One staining tray with a clothespin. Step 4. Performing the stain. Fill the 140 milliliter beaker halfway with water. Plug in your hot plate. Set the beaker on top. And then turn the temperature to high. Next, we prepare the slide for the bacterial smear. Take your deionized water and place the smallest drop possible without squeezing the dropper bulb on top of the slide. Take your bacterial culture and remove the plate cover. Sterilize your loop for 10 seconds and let cool before collecting a small sample. Press lightly to avoid collecting agarose gel with your sample. Inoculate the slide droplet with your loop. Use the loop to spread the smear to the largest possible size. This will make heat fixing easier and quicker. Sterilize the loop again before placing it aside. Now the heat fix. Using your clothespin, hold the slide over the warmest area directly above the bacticinator. When finished, the smear should have no luster and be completely affixed to the slide. At this point, your water should be at a boil. Take your slide and place it square on top of the beaker with a paper towel over the smear. Take your primary stain, malachite green, and use it to saturate the paper towel with dye. Saturate with more malachite green if the paper towel appears to be drying at any point during the staining process. Otherwise, it may adhere to your smear and resist removal. Wait for 5 minutes.
5 minutes later.
Remove the slide from the steam and peel off the dyed paper towel. Rinse the slide thoroughly with deionized water. Take your secondary stain, saffron, and use the dropper to cover the smear. Let the stain rest on top of the smear for 30 seconds.
A few moments later.
Rinse the slide thoroughly with deionized water again. Once rinsed, blot your slide between sheets of bibulous paper. And there you have it. Your finished slide. When you are done, be sure to dispose of the excess stain and rinse in the designated container. Pour the contents out of the tray into the container. Rinse the inside of the tray with deionized water. And there you have it, an endospore stain to help you explore the microscopic world.